Hi there, welcome back. Have you ever wondered how our body utilizes oxygen so effectively? And how is it that we constantly have enough oxygen in our body until, of course, something goes wrong? Explain to a 5-year-old, hemoglobin is a bus that transports oxygen to their destination. Watch this video till the very end and see how it relates to hemoglobin binding and oxygen tension. This is a special series of oxygen transport where we will discuss oxygen content, hemoglobin, oxygen-hemoglobin dissociation curve. This is Mastering Anesthesia, so let's go.
Hi, welcome back to Mastering Anesthesia. Today we're going to talk about transport of oxygen in the blood. So there are only two major ways of how oxygen is transported in the body. This is from the lungs to the body tissues. So number one, majority of the oxygen is bound to hemoglobin. This consists of about 98% and dissolved in plasma constitutes of only 2%. Let's talk about hemoglobin. A fully saturated 1 gram of hemoglobin can accommodate 1.34 milliliters of oxygen. This is known as Hofner's constant. Remember this constant because we are going to use it in our next formula of oxygen content. Next method of oxygen transport is by dissolved in plasma. It is wrong to assume that oxygen that is transported via plasma plays no important role. It is wrong. It is because this portion of dissolved oxygen in plasma is the one that contributes to the partial pressure of oxygen in the blood. So the volume of oxygen dissolved in the blood contributes to the partial pressure of oxygen because the portion dissolved in blood is proportional to the partial pressure of oxygen. Why is it important? Because this is the portion that is involved in gaseous exchange. Recall that in Fick's law, diffusion occurs along a pressure gradient. So it is this portion inside the dissolved plasma that goes into the tissues. So if we zoom into the cellular level, where we see the blood vessel with the blood inside and hemoglobin inside the blood and the tissues on the right, it is this dissolved oxygen inside the plasma are the ones that get diffused into the tissues. And once the dissolved oxygen reduces in concentration, the oxygen bound to hemoglobin will replenish the dissolved oxygen in the plasma and then continues to diffuse into the tissues. I like to visualize this concept by imagining bus carrying students. Okay, so you have the bus as a carrier, like hemoglobin, and the students being the oxygen inside the bus. So when the student's oxygen is inside the bus, there is no partial pressure exerted. But when the oxygen or the students go out of the bus, there'll be many students, many oxygen outside here. These are the one that causes tension. So the more the students or oxygen is outside the bus causing that tension, the higher the partial pressure. So now that we know that oxygen is transported in two ways inside the blood, so by adding them up, we can get oxygen content in the blood. So how much oxygen are there in our blood? We can get the value by using the oxygen content equation. Now this is a little bit daunting, but don't worry. Once you get the concept clear and understand the equation, this is pretty simple. So for oxygen content in 100 ml of blood, this can be true for artery or venous depending on the values that we put in. So we take 1.34, which is the Hofner's constant, multiplied by the concentration of hemoglobin times with saturation of oxygen divided by 100. This portion of the equation will give the value of oxygen bound to hemoglobin. And then we add the value with 0.023 times with partial pressure of oxygen. This portion constitutes the content of oxygen dissolved in plasma. Remember that it is related to the partial pressure of oxygen, so that's why we use PaO₂ here. I'm just going to re-emphasize here again. The green portion of the equation shows how much oxygen is bound to hemoglobin, and the portion in blue shows the content of oxygen dissolved in plasma. Recall 1.34 is the Hofner's constant, true at 37 degrees for adult blood. This is the concentration of hemoglobin, and this is the percentage of saturation of hemoglobin. 0.023 here is the solubility coefficient of oxygen in water, and PO2 is the blood oxygen tension measured in kilopascal. So now that we have gone through the equation and understand it somehow, let's see how this formula works. Alright, so we are going to take CaO2, the A here will stand for arterial blood. So you have a typical value of hemoglobin about 15, saturation of oxygen 97%, and PO2 of 13 kilopascal. Now to change the unit to millimeters of mercury, you have to multiply whatever the value here with 7.5. So 13 kilopascal will give us about 98 millimeters of mercury. So putting in the values inside the equation will give us 1.34 times 15 times 0.97 plus 0.023 times with 13. This will give us a value of 19.5 plus 0.3 and a sum of 19.8 mils. This is the oxygen content in 100 milliliters of arterial blood. In contrast, we take the venous blood right now. Okay, so SaO2 of venous is about 75%, PO2 of 5.3 kilopascal or about 40 millimeters of mercury. So putting these values inside the equation will give us 15.08 of bound hemoglobin and also 0.12 of oxygen dissolved in blood will give us a total value of 15.2 milliliters. This is oxygen content inside venous blood. Here I want to highlight how greatly the hemoglobin contributes to the overall oxygen content in the blood. You have 19.5 compared to 0.3 oxygen carried by the hemoglobin as compared to dissolved in plasma. And in the venous blood, about 15 milliliters are carried by the hemoglobin and only 0.12 of oxygen is dissolved in plasma. Okay, so we've talked about oxygen transport. Now, what about oxygen storage in the body? Hint, not a lot. So when the person stops breathing or apnea, the body will rapidly goes into hypoxia. So oxygen can primarily be seen stored in lungs, blood, as well as the muscles. For the lungs, they are stored in the functional residual capacity. In the blood, bound to hemoglobin and dissolved in plasma, like we've discussed earlier, and myoglobins in the muscles. And recalling from our previous calculation, we have calculated about 20 mils of oxygen in 100 mils of arterial blood and about 15 mils of oxygen in 100 mils of venous blood. So let's project these values to a healthy 70 kilogram adult man with a typical blood volume of about 5 liters. So they will have about 850 mils of oxygen in their blood. In myoglobin, it can store up to about 250 mils of oxygen, and the functional residual capacity of the lungs can carry up to 450 mils of oxygen. So when you add this all up, you have about 1,550 mils of oxygen contained in the whole body. Like I said, not that much. Why, you may ask? Because even the resting oxygen consumption of the body is about 250 mils per minute. So oxygen will be depleted within a few minutes. Therefore, in few minutes of apnea can have serious devastating effects caused by cellular hypoxia and also hypoxic damage. And this can be made worse in conditions where there is increase in oxygen consumption or reduce in oxygen carrying capacity. This includes sepsis and states of oxygen consumption increased, and also anemia and carbon monoxide poisoning, where there will be a decrease in oxygen carrying capacity. Now that's been taken care of, let's talk about hemoglobin, the primary vehicle of oxygen transport in the blood. They are contained in red blood cells. They are a large iron-containing protein. There are many different types of hemoglobin which can be divided into physiological and pathological. The most commonest form of hemoglobin found in blood, is about 95%, is HbA. They have quaternary structure with 4 polypeptide chain globin subunits. So they have 2 alpha and 2 beta. This makes the HbA. They are arranged in a tetrahedral manner, some sort of like the pyramid where it's concentrated in the middle. Each globin chain has one heme group, which is an iron-containing porphyrin ring with the ion in Fe2 plus form or ferrous form. The globin chains, 4 of them, are connected via weak electrical forces, and one hemoglobin molecule can bind up to 4 oxygen molecules. So when we talk about hemoglobin in the body, it exists as 2 forms, either deoxygenated or oxygenated. Deoxygenated are called deoxyhemoglobin, and it is in a tense conformation, whereas in oxyhemoglobin, it is in a relaxed conformation. So the primary function of hemoglobin is to bind oxygen and transport the oxygen. So when it does not have oxygen, which is deoxyhemoglobin, it is tense, it needs oxygen. Okay, but when they are oxygenated, it fulfills its purpose of holding oxygen and delivering it. So it is more relaxed because it has already gotten the oxygen. In a sense, I would visualize oxygen as tranquilizers for the hemoglobin, right? So the oxyhemoglobin has oxygen, so it's relaxed, and the deoxyhemoglobin needs oxygen, so they are tense. Now for the diagram. Oxyhemoglobin has 4 globin chains, 2 alpha and 2 beta, with 4 heme groups. Oxygen attached to the ferrous group there, so it has this relaxed state where all the alpha and beta subunits are close together. And then for the deoxyhemoglobin, so you have 2 betas, 2 alphas, and these beta subunits are separated, they are far apart due to this presence of 2,3-DPG. So you have these heme groups, and no oxygen is attached to the heme groups of the hemoglobin. So from this tense state, when oxygen binds to the hemoglobin, 2,3-DPG will detach itself from the hemoglobin. So from the tense state, it becomes the relaxed state of oxyhemoglobin. Whereas when the oxyhemoglobin from the right side releases oxygen, gains 2,3-DPG, converting it from relaxed conformation of oxyhemoglobin to a tense conformation of deoxyhemoglobin. A unique feature of hemoglobin that we need to know in order to understand oxygen-hemoglobin dissociation curve later is that it exhibits cooperative binding. What it means is that one binding eases the binding of the next subsequent bindings of oxygen. So increase affinity of oxygen to hemoglobin with every successive oxygen binding. Let's see what happens with the first oxygen molecule trying to bind to the heme group. The beta subunits are far apart in a tense form, yeah, remember? So strong electromagnetic forces need to be overcome. So once it binds to it, the beta subunits come closer together. Then comes the second oxygen molecule trying to bind to the hemoglobin. So the hemoglobin has a higher binding affinity. The beta subunits are already closer together. And on the third and fourth binding, it becomes easier and easier. So by the time it has reached the fourth oxygen trying to bind to hemoglobin, it has become about 300 times easier or higher in infinity as compared to the first binding of oxygen to hemoglobin. So there you have it. Thank you so much for watching. I hope you enjoyed this video. Please stay tuned. There will be more parts to oxygen transport, and see you in the next video.
Thank you for watching. If you find these videos helpful, please hit the like and subscribe button to show your support. Comment down below what topic you want us to cover next. Till next time, see you in the next video.